required for adipogenesis, and then just observe in many conditions in different genders. When do we see adipogenesis? When we don't see adipogenesis, and which factor is important and which is not that important. So, but bottom line, I think so. We all come to one conclusion is that the adipogenesis of APCs in healthy adult is in a very low rate. So basically, adipocyte turnover is low in young adults, especially in mouse. Humans, like there's early papers 10% a year. So we think also we have many evidence. We and the other labs is that only adipogenesis in a young adult are only activated by metabolic challenges, which is prolonged hepatitis feeding or cold exposure conditions like that. So, so going back to this recent work that we did is that because we was doing lineage chasing for different conditions, we kind of miss the aging because aging just takes a long time. So then we look at, first we look into the Y type, right? This is really just towards mid-age. So if you, firstly, we compare the body weight of male and female, and we see actually the male gain a lot of weight at 1 year old. And if you look into the body composition, you can see that the increase of body weight is majorly contributed by the increase of fat mass and also fat percentage. And lean mass actually increased a little bit, but by percentage-wise, because of the increased fat, so the percentage lean mass just decreased a little bit. And if you look into different type of fat depots, gonadal fat is a typical visceral fat we use to study in mice, and you can see that it's really increased by a few folds, and also subcutaneous fat are increased in their mass as well, but it's just not to the extent as the gonadal fat did. So then this is like 1 year old, like a mid-age, early aging male mice, Y type, and metabolic cage studies. You can see that those mice at mid-age, they just have reduced oxygen consumption, reduced energy expenditure, slightly reduced activity, slightly reduced food intake. So this dropping oxygen consumption, energy expenditure suggests that the aged mice have significantly lower basal metabolism. So this is really similar to aged humans. And with that, this is not very surprising. So this is just a child that fat mice at 12 months old. So compared to the young mice, they really do worse at GGT, the glucose tolerance test, or also the insulin tolerance test. So this means that so with mid-age, with the fat expansion, the mice gradually become accumulating those metabolic disorders regarding to glucose insulin sensitivity. So so then we went back to our lineage chasing model. So so this is a negative lineage chasing with a double chaser mice. So this is a combination of 3 mouse lines. So you have the adiponectin promoter-driven RDTA. So adiponectin is specifically expressed in mature fat cells, and then you have a TRE promoter-driven CRE, and you have a ROSA 26 and ROSA 26 tomato and GFP. So at a basal level, tomato is always activated. So when you cross those lines together, whatever every single cell in this mice are tomato positive. So if you put the mice on a doxycycline diet, and then the doxycycline will activate RDTA, so that RDTA bind to this TRE promoter and then drive CRE expression. So the CRE will then just cut the flux region, any flux region. So because the tomato sequence was fluxed, so it will be cut off. And then you will have MGFP directly under the ROSA 26 promoter. So you're going to turn on CFP expression specifically in mature fat cells. So and if we so this turning on is permanent for this cell, and then we can remove doxycycline and then chase. So what we did was to chase for 3 months, another 3 months, another 3 months until so from label at almost 3 months old, and then observed until they're 1 year old. And you can see that as we reported before with other reporters is that at the first 3 months, there is really no very low level of adipogenesis. So how do you read this image is that so paralipin stains for all the mature fat cells. So it's just the stains for the lipid droplets. And GFP stains for the fat cells that was labeled at this stage. So at 6 months, you can see almost all the fat cells are labeled, so which means that the adipogenesis is really not really contributing. And starting from 9 months old, you start to see really a lot of negative, a lot of live adipocyte that is actually GFP negative, which means that those cells are generated during the chasing period. So when we're labeling at this time, they were just a precursor, so they were not really mature adipocyte. And at 12 months, we just see more of adipocyte are generated during this stage. So by quantification, you can see that this is really a sudden increase of adipogenesis around 9 months old and continue to increase at 12 months old. And at the same time, we did observe adipocyte hypertrophy, which was reported before because it's easier to observe if you just do a HE staining in mid-aged mice. You can see that the fat cells are definitely bigger compared to the young mice. So that was negative lineage chasing. So we also did positive lineage chasing. So this is very similar to the adipotracer mice. We just switched the adiponectin RTA mice to PDGF receptor alpha RTTA. So PDGF receptor alpha is one of the few markers that right now we generally believe to be marking most of the APCs. So if we can label the APCs at the same time, like when they're young, a little bit before 3 months old, and then chase for the same 3 months, another 3 months until they're 12 months old, we can see if those labeled progenitor cells will differentiate into fat cells. So you can see that similarly at 3 months old, they're just progenitor cells. 6 months old, they're also progenitor cells, but starting from 9 months old, they become to have this mature adipocyte structure, the GFP positive cells, which indicate that those progenitor cells had differentiated into mature fat cells during this time frame. So and this quantification is quite similar. So we see a sudden increase at 9 months old, and then a dramatic increase at 12 months old. So both positive lineage chasing and negative lineage chasing shows that APC actually adipogenesis of APC is unlocked during mid-age in the male mice. So we then asked a question. So do the APCs in aged mice cell autonomous have increased the adipogenic capacity, or is that because they are mid-aged mice, if we just give them any young APCs, this aging environment, it will just trigger adipogenesis. So the experiment we did is, so we, as I mentioned, we have row cell of MTMG mice. So at the basal line, those mouse just every single cell are tomato positive. So then we purchased another CAG EGFP mice. So every single cell in this in these mice are just green. So we did age like a 12 months old tomato positive mice and a young GFP positive mice, and isolated APCs, mixed the same amount of APCs, and then transplanted to the young white type recipients. So a few weeks later, you can see this is the 3D imaging of the transplant. So tomato positive cell, you can see the round shaped adipocytes. Tomato positive cells are different. Actually, the age when they're really in a competitive environment, the age APCs starting from the same number actually give rise to much more mature adipocytes, even in a young environment, right? So this is just a close look at the transplant. So this, if you take the fat pad out, so this is actually the matrigel and the cells transplanted. And if you do a sectioning, you can see that GFP positive cells, some of them are really have this round shape. So they are a double, mature adipocyte. So they're from the young APCs, and the tomato round-shaped adipocytes are from the aged APCs, and paralipping shows that a lot of them are actually showing mature adipocyte features. So this is just the quantification of a few transplants. So from here, we think it looks like it is cell autonomous, right? So and then the reviewer actually ask for so what if the mice who purchased the tomato positive cell tomato line just has more adipogenic capacity. So what we did is just switch the colors. So this time we transplanted aged APCs as GFP positive cells and young APCs as tomato positive cells, and you can see that and we got similar conclusion. So the aged APCs then differentiated into more adipocyte, mature adipocyte than the young tomato positive cells. So then we have to answer we have to answer the other question is that the aging environment, right? So we did then isolate the young and so young APC and aged APCs to see, this is into young mice. So I from the gonadal of young tomato mice and then transplanted those cells into one side of the young recipient, and then isolating APCs from the gonadal of aged tomato mice and transplant those cells to the other side of the young recipients. So if 2 weeks later, again, you can see that the aged APCs actually generated into like a much greater number of adipocytes. So this is still similar to the previous experiment. So it's the APCs are cell autonomously gaining more higher adipogenic capacity. So this is just the 3D imaging of the transplants. So yeah, so excuse me. So this one actually is to answer the second question, the aging environment. So we then did 2 colors, just repeating the same experiment. We isolated the young APCs and then transplant those cells into young recipients or age recipients. And you can see that regarding to if they're in a young environment or if they're in an age environment, they actually generate into same numbers of adipocyte, and this is the same in the GFP positive mice. So this indicates that actually the aging environment at that time. So we, of course, the APC are remodeled by aging, right? It's just at that moment, this aging microenvironment doesn't really trigger the adipogenesis of young APCs. So then when we were doing those transplantation experiment, at the same time, we were just seeing like numerous publications on single cell sequencing of APCs. So this technology had really given the field a great opportunity to reveal APC heterogeneity. So forgive me if I didn't put your favorite especially were also very interesting to see Dr. Souls from UC Berkeley. So they published a paper in Development Cell and showing the discovery of aging-dependent regulatory cells that emerge in subcutaneous fat that is inhibiting adipogenesis. I think that kind of explain why we actually didn't see much adipogenesis in the subcutaneous fat. So we saw actually massive adipogenesis only in the visceral side. So with that, so we just can't stop thinking of we really need to do a single cell sequencing to see what's really going on with the aged APCs. So we did 3 and 3, so 3 young mice and 3 aged mice, and isolate their APCs. This is actually just link negative stromal vascular fraction cells. So you can see that most of the cells are progenitor cells. And if you just single out the progenitor cells, you can see that young and age are kind of there's a dramatic shifting in transcription like gene expression, so indicating that age kind of remodeled the APCs. And if you look into individual clusters, we was able to make find 5 major clusters. So we kind of, we read a lot of publication at that time, so we try to name them similarly. So they have adipocyte stem cells, so ASCs. This is similar with Patrick Sell's science paper, and also intermediate adipocyte progenitors, and also we found committed pre-adipocyte 1 and 2. So all those 4 populations share a lot of similarity with published clusters, their top markers. But at the same time, it was like very exciting for us is that we found this age-specific committed pre-adipocyte. So we named them CPA population. So if you separate young and age, you can definitely tell that the CPA is very unique to the age populations there. So then later on, we actually generated the mice. So this is THPS1 GFP mice. So THPS1 is one of the top markers of CPA, especially in mice. It works really well. So we were able to really enrich to see how many CPAs do we see at different ages in different fat depots. And you can see that really in line with the lineage chasing data, we see actually no CPAs in 3 or 6 months old mice, and we suddenly see this population at 9 months old, and then we see a peak at 12 months old. And then for the gonadal-fat, there's a sharp drop of this population at 18 months old. I think this is usually people consider about aging, right? 18 to 2 years old, and this really pairs with our kind of the first review image where I show you, like this pairs with the advanced age. There's then a lean phenotype. So this the adiposity is really a mid-age specific phenotype, and we see similar population and similar trend in the subcutaneous fat. It's just in a much lower number there. So then we did a trajectory assay. So this is computational, and you can see that the CPA is really leading the stem cells to a new trajectory. So usually it goes to the intermediate cells and go to CP1 or CP2, but it actually also goes to CPA. And so then the postdoc 1 actually did this experiment. So she actually isolated the ASCs from the aged mice and then transplant those cells into young mice, and this is only after a few days. You can see that so young and aged ASCs then transplant to young recipients. You can see that the young APCs stays there, right? They don't really go down the lineage to become LEAF-R positive. So LEAF-R is actually another top markers of the CPA population. And then in the aged ASCs, even though they were transplanted to the young recipients, they actually actively go down to the lineage. So this indicates that the aged APCs are very actively really just proliferating and going down to the lineage and then really generating into new adipocyte. So going back to the markers, right, I mentioned the CPA has 2 top markers, which is THPS1 and LEAF-R, and also we were able to see that all APC express PDGF receptor alpha, and also the ASCs express the markers that reported by initially by Patrick Sell and then many other labs. So it's CD55 and DPP4. So those are in mice, and we were of course very interested in. So do we see that in human, right? So luckily, Cedar Hope has this islet transplant group, so they constantly receive pancreas from the diseased donors. So we would just get the peripancreatic fat tissue, so which is considered to be a typical human visceral fat, and that's the only fat we can get. So we as we get sample, we just send out for single cell sequencing. So we did sample 5 humans, and so I won't look into the details of the clusters. But bottom line is, if we look into the top 20 markers of ESC, we can actually find this population in human visceral fat, and then also if we use the top 20 markers of CPA from the mice, we can really distinguish this population in human as well. So and then if you look into individual genes, all the progenitor cells that are expressing PDGF receptor alpha, like we saw in mice, and also you can see that the ASC's population express the typical marker PS16, and the CPA marker, CPA cluster express the top marker LEAF-R. So LEAF-R is really shared between mouse and human. And so we then used LeafR to do flow analysis. And once we receive another human donor, so those are all male, and you can see a trend, right? So it's really hard to tell with this limited number. So when they are really young, you can see the percentage of LeafR positive APCs are really around like from 2 to 10%. But when you get the samples from mid-aged male, and this can increase to 20 to 50%. So we think maybe this enrichment of CPAs in visceral fat in human is also contributing to the age-related adiposity. So going back to the mouse study, the single-cell sequencing data. So we firstly we compared aged stem cell, edible stem cells with the young ESCs, and you can see that one of the top enriched pathways is TGF beta signaling. So this is also reported by Patrick Cell that is important for promoting the proliferation of APCs. So we think maybe the aged ESCs are proliferating more. And for the CPA population, it was really hard for us to compare because the young group just don't really have, only have like a single digit number of CPAs. So we just look into just what pathways are enriched in this cluster. And you can see that Adipogenesis is one of the pathways that's enriched, and also like JAK-STAT3 signaling, which we're gonna talk about later. So we wonder, can we do some in vitro studies to validate? So if you work on adipogenesis or adipose tissue biology, you may have an impression that when you isolate the SVFs from the older mice, right? They were really hard for like we compared to young mice. They either have similar adipogenic capacity or they even have lower. And this is similar with human. A few human data, human paper reported that if you isolate human SVF out and differentiate it in a dish, the aged human often like really give you a lower adipogenic rate. So we did similar 2D culture and adipogenesis, and we have the same observation. So either we have no difference, mostly no difference of the young and age. So then we thought about like because this 2D culture protocol really requires a long culture and conflict growth until they are confluent and growth contact and really they kind of change a lot of the molecular signatures of those APCs. So we eventually figured out a new method of 3D culture of the SVFs. So long story short is that those are just a 3D culture in a matrigel. So we were able to add a adipogenic cocktail only 16 hours post-seeding, so as early as possible, as we want to keep them their features as they were still in vivo. So comparing aged ESC and young ESCs, you can see that you don't really have an increase of adipogenesis, but you have you end up with more numbers there, more total cell number or more total double cell number, which kind of just indicate that those cells are proliferating more. And if you compare same number of age CPAs and young CPA1, so young CP1 is really the cluster in young mice that shares the most molecular similarity with the age CPA there. So you can see that you have a really a one-fold increase of the adipogenesis rate, and also dramatic, like even like a 4-6-fold of increase of the total cell number and the double cell number. This may indicate that the CPA population not only have increased proliferation, they also have increased adipogenic capacity. So from this 3D model, we can see that actually we successfully mimic what we saw actually in vivo and appears with mid-age increase of adiposity. And then we thought about how about we just do the transplantation experiment again, because the first data I showed you is just the total APCs. So here we enriched enough numbers of the age CPAs and then enough numbers of young CP1s. So we just transplant those cells into the same young mice, and we compare like how a few weeks later how they behaving regarding to adipogenesis. So you can see that this is really shocking to us. If they start the same number of CP1 and CPA. The CPA mice actually gave rise to a fold increase in the transplant size and also the adipocyte number. So again, it shows that if you enrich the CPA, they just have even higher adipogenic capacity and really actively undergoing adipogenesis in the in vivo setting. So last part of this presentation is about the molecular mechanism, right? It's obviously very important. Then we look into the few markers. So LIFR has a full name of leukemia inhibitory factor receptor. So we searched the literature and found that this LIFR signaling can control several cellular processes, including proliferation, differentiation, and survival. And it's important for immune regulation, also like in cancer. But its role and in APCs has really not been studied. So firstly, we did SHRA of LIFR just to knock down the expression of LIFR in isolated CPAs. So you can see that if we knock down LIFR, you have a dramatic reduction of adipogenesis in a 3D culture, and also we transplanted those treated cells into the same recipients as the left and right side, and can see that this CPAs with all LIFR really has a dramatically impaired adipogenic capacity. So what if we overexpress, right? So we overexpress LIFR. Even just in young APCs, we didn't really isolate subpopulations. Just simply overexpressing LIFR, you can see that you can dramatically increase the adipogenic capacity. This is in a 3D in vitro culture, and this is the transplant of different sites of young recipients. So again, if we overexpress LIFR, actually promote adipogenesis. So lastly, we were able to find known LIFR inhibitor EC359, and we used again in the 3D culture environment or in vitro transplantation after treatment. And you can see again, if you inhibit LIFR signaling, you have dramatic reduction of the adipogenic rate. And this is when you treat the mice and treat the cells and then transplant to the mice, you have a dramatic reduction of adipogenesis of the CPAs. So I think this is the last data. No, this is we have two more data slides here. So if you look in the literature, LIF and LIF receptor, the downstream is STAT3. So firstly, we look into the protein, the phosphorylation of STAT3. If we use the LIF inhibitor in the CPAs, actually you have a dramatic reduction of phosphorylation of STAT3. And then we were suggested by Yuhua at City Hope to use a few of STAT3 inhibitors. So we use static and NAPA, and you can see that. So this is to test whether STAT3 is required for our adipogenesis, or especially for the aged aged APCs here. So those are actually not enriched. Those are CPA. Those are CPA and CPA1, I believe. And if we add STAT3 inhibitor, you can see that there's dramatic reduction of the adipogenesis rate of the aged CPAs. But the young cells, they just they start with a comparatively very low adipogenic rate, and they don't really respond to the STAT3 inhibitors, which means that STAT3 signaling is kind of specifically inquired for the age of adipogenesis. So lastly, we used this EC359 as a LIFR inhibitor. We used it from 9 months old towards 12 months old. So this long term of treatment really ended up with a reduction of the gonadal fat mass. And this is what we are showing all the time today, is that we see a gonadal fat, actually the visceral fat has dramatic adipogenesis and contribute to the increased fat mass gain. And at the same time, we didn't see a huge difference in the subcutaneous fat, again validating that for subcutaneous fat, adipogenesis is not really a dependent factor for them to increase their mass during aging. So with that, this is just a summary. So firstly, as in humans, mice have really similar age-related visceral adiposity. This is mid-age, really mimicking human sarcopenic obesity. And with lineage chasing, different lineage chasing mouse models, we saw that massive adipogenesis is triggered during mid-age and early aging. And then with in vivo transplantations, we found that APCs from aged mice actually so autonomously have a greater adipogenic potential, which was not really found in traditional 2D culture system. And then with single cell sequencing, we found that aging really it generated this age-specific committed pre-adipocyte population. So we named them as CPA population. And this CPA population, both in vitro and in vivo, they have just a very striking high adipogenic capacity. And lastly, we found that the LEAF-R and STAT3 signaling are really required for CPA adipogenesis. And again, I want to emphasize that the whole phenomenon is really time dependent. So it's a mid-age, early aging phenomenon when the mice become much older, then the thing could be quite different, which is also what we are interested in studying, kind of the future plan. So with that, I would like to thank my lovely members. So the whole project was a collaboration of our lab. So the postdoc Guan Wang did all the experimental studies, and we were collaborating with Yang Xia at UCLA. So Gao Yan was a student in Xia's group, so she did all the single cell sequencing data analysis. And also, I'd like to thank our collaborators at State of Hope and our core facilities. And I want to really thank Bengla Shao, who is also the panel member today. So he actually helped a lot with our experimental design and writing the discussion of the manuscript. And also, Share, Rana Gupta, and Yiju are really long-term supporter. And and also the funding agencies. So I want to just especially thank my PG mentor, Yung Liu. I don't know if he's here today. So so he has been really supporting me for since I joined his lab, and I really helped me to be confident in science and be finding the excitement of doing science. And I think I will not really have a lab and continue to do science without his help and encouragement. Lastly, so we just secured a few fundings recently, so we have postdoc positions available. So please just email me if you're interested. And also, Guan Wang, as the first author of this paper, she actually accepted an offer from Taikang Shimi Xie at Wuhan University. So you're gonna see her a lot in the future. Thank you so much.
Thank you, Annabelle, for the great talk to share your story on the adipose aging new findings. So now let's pass to Julie.
You take over for the question Q&A session.